fit parameters given the structure. And so you try to be as accurate as you can with your training data and hope that holds up on the test data. But what all your collaborators really want is a causal model. And we know very well that there are many different true causal models that could give rise to the same Bayes net we just saw. There are many different Bayes nets that might capture the same probability distribution is another way to describe this. So to actually get at the causality, people looked at knockout experiments or just doing Granger causality with time series data, relying on the fact that we won't be able to build a time machine. So we're expecting if A causes a future B, or if A is a good predictor of a future B, there's a better chance that it's causal rather than B causing A, and so forth. So around this time, I started at the University of Wisconsin in 1999 and started working with Fred Blattner's group, which had sequenced E. coli, which at the time was big news. It's a super easy sequencing problem now, but back then it was a big deal. And they taught me about operons, that you get a run of genes that get transcribed together and then get translated into separate proteins. And they could show examples of this kind of operon behavior as I'm illustrating here. And so we wanted to model the whole gene expression regulatory network in E. coli. And my student, Irene Ong, although others had, Daphne Kohler, Kevin Murphy, others had proposed using dynamic Bayes nets for this, Irene was the first to apply dynamic Bayes nets to time series gene expression data. And the network looked like this, where we had an operon model we had already predicted that gives us the top to bottom arcs. And then we want to learn the other arcs, additional arcs that are going right to left. And so this was state of the art for a while, and then everyone moved to conditional random fields. But I want to stop and say that a big problem we encountered is that this worked fine using the EM algorithm, which in this context is essentially the Baum-Welch algorithm you would use for hidden Markov models. This worked fine until you had really long time sequences. So we published a paper with time sequences of 4 or 5 steps. And then we had lots of people contacting us with data of 25 or 30 steps, and it just failed miserably. Now, computationally, a recurrent neural network or a transformer or one of these methods would work really well, really efficiently. But we didn't know about that at the time. And so just as a practical matter, that would be a reasonable approach now. But I want to say it's more than just a practical approach. Also, around 2005, 2010, there was a big shift. While the deep learning revolution was just starting, there was a big shift from Bayes nets to Markov nets, undirected models, conditional random fields, because you could represent them with lots of features, even far-ranging features like this variable way back here is true or is bigger than some value, and this variable way up here is false. You could look far away with the features of a CRF, just like you do now with a transformer, but you still had the computational efficiency problem. So CRFs, you could imagine the very same structure I just showed you, but make the arrows undirected. And there's a big caveat, which is that you have to normalize at the end. Your values, your answers you get, your probabilities are not normalized as they are in the Bayes net. I'll say why that's a big problem in a minute. The advantage of this approach is that features can capture long-range dependencies, and you have to do gradient descent, but it's convex, and the gradient is really simple. For each of your features, the amount you just adjust the weight on that feature by is just the difference in the probability of the feature given your input variables compared to your inputs and outputs. And so the gradient has this really simple form, and you're just going to look at that across all of your features. The big disadvantage is computing the normalization constant, which is 1 over the partition function, it's called, is terribly inefficient. You basically have to look at every possible grounding or value for every one of your variables. So you're looking at exponentially many combinations over your number of variables. So now, of course, in practice, everybody has left, or many people have left CRFs and are working with deep neural networks. But it was tough enough for me to shift from logic to probability and statistics, and then shifting to neural networks, where all we got was a probability of y given x out at the end, was a big shift because you're ignoring most of the model. A Bayes net or a Markov net or CRF is giving you the joint distribution over inputs x, hidden variables h, which is most of what's going on in a deep neural net, and outputs y, or in a CRF, the conditional y and h given x. And much of the action is in h. So that's background motivation for what I'm going to talk about the next 15 minutes. And then the rest of the talk will be implications of that. Let me stop and see if anybody has a question now. Feel free to just shout it out if you have one. Okay, so that's big picture motivation. Several of the folks here are involved or have been involved with the National Human Genome Research Institute's program. It's kind of their follow-up to the ENCODE program called Impact of Genetic Variants on Function, or IGVF, the first paper that came out yesterday. And I've been fortunate enough to be involved with just one of the many grants that's in this program. And we're using this. This funded a lot of the work I'll talk about today, so I should acknowledge that. But the opinions are mine and not NIH's. But the motivations come from this program largely. IGVF uses lots of probabilistic graphical models, especially for pre-processing, but it also uses lots of deep neural networks. And if you wanted to glue those together and fine-tune them into one global regulatory network model, it's unclear how you would do that. So this would be an example of one of the plate models or probabilistic graphical models where we use Hamiltonian Monte Carlo to do inference. And then there are also a number of neural network models involved as well. But the goal is to glue all these together into this much bigger model where we can go from genetic variants over at the far left via the various changes those induce to phenotypes such as change in expression across on the far right. And there are different components that have been built by us or by others that can do these different portions. And one of the implications of what I'll talk about today is that we can glue these components together, even if some are neural nets and some are probabilistic graphical models, because we can view the neural nets as themselves proper probabilistic graphical models. And so that's what I want to talk about. One downside is then when you want to fine tune, what you've got at the end is a probabilistic graphical model, not a pure neural net. So you do have to use Hamiltonian Monte Carlo or some of these other slower techniques to do that. So the motivation is there's a lot we can do if we can understand the neural net is giving us P of Y and H given X, and not just P of Y, not just the black box machine that gives us P of Y given X. Another thing we can do is do inference not just from inputs to outputs, not just forward inference, but we can do backward inference or inference anywhere, put the evidence anywhere, ask about query variables anywhere else in the neural net using, for example, this Hamiltonian Monte Carlo that's based on the theoretical result. Okay, so these are some of the motivations for what we'll talk about now. So we got interested in this at first when we observed that in the special case that I'd been teaching for 30 years of neural nets where the activations are sigmoid, so let's stick to that for now, each node is just a little logistic regression model. So in my colleague Larry Karen's words, a neural net can be thought of as just stacked logistic regressions. This fits the formal definition of a Bayesian network, which is just a directed acyclic graph where every node has a conditional probability distribution conditioned on its parents. It's just each of these is going to be a little logistic or sigmoid. It also fits the definition of a Markov network because we're going to normalize this at the end. And so we're going to take, we're going to view this any edge as a feature where the feature is true if and only if the node on each side of the edge is true. And that gives us a little table like this in the Markov net where when the feature is true, we get e to the power of the weight as compared to a 1, which if you normalize, just gives you the sigmoid of that weight as the probability that this edge is true or that this node is true. It turns out, though, if you dig into the details of this and compute the gradient that's used when you do gradient descent in a neural net, whether it's general gradient descent or as we typically do now, stochastic gradient descent or atom or so forth, the gradient is actually not correct with respect to either of these. The gradient's different with respect to each of these and different for the neural net. So what's going on in this neural net? One way to think of this is it's using an approximation. It's just doing something different. So can we characterize this approximation or can we characterize if anybody has a question or comment? Okay. So the whole idea that the neural net is doing just logistic regression, I left out the one bit, which is neural nets started to have their best successes when they moved from squared error to cross entropy error functions. And I think that's not an accident because that's where a little logistic regression model gave you this idea easy. At least for us, it turned out to be really hard. So I'm excited that I can show you the result. The first big issue with what we think is going on, or another way to say it is if you try to show that Bayesian network, that the gradient is the same as the neural net gradient, if you try to show that, the first big problem you run into is what's actually happening in the neural net is, if I go back a little bit, H3 and H4 here, they both have arrows coming into them from H1. H1 is a parent. But in the neural net, what's actually happening in effect is that H3 and H4 each have their own copy of H1 and everything goes between these variables. What we've essentially said is that H3 and H4 have the same conditional distribution conditioned on the input, but they're not tied to have the same value. Showing this as a of tree structure graphical models. They're kind of like the one case where we can do everything efficiently in Bayes nets or Markov nets. For example, that partition function, we can normalize at every node if this is tree structure, and we don't have to wait until we've done the full computation. So some practical downstream results are that we can now glue PGM and DNN components together as long as we keep in mind what we really mean by this DNN component, that it's really a PGM. Another downstream impact is that each of these nodes in the neural net is standing for a big sample in that tree-structured Markov net, and L is telling you roughly how big of a sample that is. And L translates in the Hamiltonian Monte Carlo algorithm to just saying how much noise or randomness we're incorporating when we sample that value. So if you take that neural net and you do Gibbs sampling in it, like I said, you're not really getting the same thing, because it is a Bayes net, but it's not a Bayes net that's equivalent to the Markov net. But Gibbs is also very inefficient. And so Jeff Hinton, others tried Gibbs years ago, and it just did not work very well for both reasons. It's not equivalent to the neural net, and it's woefully inefficient. Now in HMC, you're sampling continuous values, which are the aggregates from all these many binary valued nodes, and your L controls how much randomness you're having, or you can calculate your L as a measure of how big you believe your samples are, or know your samples to be. I'm not saying you would ever actually build this model. If you don't like this model, you can argue with me that it's not a great model, but if you're using a DNN, it is what you're using. And so that's the point of the theorem, that you're already using this. So have a look at the, we're waiting for, like everybody else, we're waiting for a NeurIPS result on this, and hope to have that soon. But the paper's up on archive if you want to see the proof right now. And I don't want to keep you waiting for that. And then there are some limitations here. One is this assumes you're using cross entropy. I don't plan to do anything about that. We're committed to cross entropy as the error function. But I also, to do the proof, we used sigmoids, and in practice we know we use a lot of other activation functions. Jeff Hinton had a great paper years ago arguing that ReLU itself can be seen as a sum of infinitely many sigmoids, and so this generalizes quite naturally based on that to ReLUs. But more generally, if you have any other activation, I can't help you if it can go negative. But as long as it's non-negative, because this is a Markov net and it relies on normalization, there's no problem theoretically. If we want to equate the actual training that you're doing in the neural net, then we have to think of the batch normalization or the layer normalization that you have to do to make these things work as in fact an approximation to the proper node by node normalization that you would do in the full Markov network. So I do want to give credit that my collaborators on this were Boyal Lee, Alex Thompson, Hussam Nassif, and Matt Engelhard. Boyal implemented the HMC, which is up and available on GitHub if you want to play around with it. Alex did the hardest parts of the proof. We all had lots of comments and discussions, and years actually of discussion around this, and everybody put in a lot of work, but Alex got us over the hurdles with the hardest parts of the proof. Do play around with HMC. Alex and Boyal are also looking at message passing algorithms, other approximate algorithms in Bayes nets and Markov nets. But where I want to go next, I'm sorry, I've got another typo in here. Where I want to go next is some downstream potential implications of this. And I want to focus on two topics kind of hand in hand. One is, if we can all agree we've made our peace now with treating neural nets as proper statistical models, and we're happy to use them. My big goal for whatever time is left in my career is to have a foundation model for health. If you're familiar with the term of foundation model, LLMs like GPT-4 that are trained on enormous amounts of data, trained once, and then you tailor them to your task with a dialogue, with an interaction that doesn't further change the weights. That's a foundation model. You may know the biggest topic probably right now in AI for biology is protein language models like AlphaFold 3, and I can talk more about those. And those, you can have foundation models in that space. I think we need a foundation model for all of health. I'll talk about some of the barriers to that and then how we can get to that. I will say, I forgot one limitation on our previous result that we haven't addressed yet. This is not yet extended to transformers. From support vector machines, you know that you can view a kernel as expressible in terms of features. And so we believe this extends to transformers, but we haven't done that piece yet. But if you'll give me an IOU on that one for now, then I think theoretically we've made our peace with moving to this foundation model for health. And then I want to say that almost every time I've worked with real healthcare data, the clinicians, and certainly in that earlier work, the biologists want causal models, not just predictive models. We moved from Bayes nets to time series and dynamic Bayes nets because of the need to be more causally accurate. And I think that ties into what we want in a foundation model. So let me stop again and see if anybody has a question or comment. You're taking pity on me with my hearing aid being out. Okay. So if you don't know about protein language models, be sure to go look at these. They're pretty fascinating, pretty incredible. From a machine learning perspective, they're really not any different from GPT-4, from the LLMs. But as somebody who myself, as a postdoc, worked for 2 years on the protein folding problem and finally succeeded in getting a paper out that improved the state of the art by 1 percentage point. I marvel at what AlphaFold 3 can do. And my colleagues at Duke, Pranam Chatterjee and Rohit Singh, tell me that you don't even have to first use AlphaFold 3 to predict the structure and then try to predict protein-protein binding or protein-ligand binding. You can go straight to, from a pre-trained foundation model, straight skipping the structures of the proteins you're interested in, straight to protein-protein binding, protein DNA binding, protein ligand binding. Of course, there's the potential for now the kinds of technologies that are being extended to things like generating video and generating small molecules and generating potential drugs. There's the potential to go far beyond just proteins. So, of course, to review some of the key steps that led us here, there were new architectures like transformers, much larger data, enormous processing power, certainly some improvements like using cross-entropy error function, using ReLU and other activation functions, the whole idea of self-training that started with AlphaGo and then AlphaZero. But I would argue maybe the biggest step in all of this was when the community decided to stop treating every natural language processing task as a different task and try to treat them as one unified task, to have a foundation model like GPT-4, ChatGPT for all of these different tasks. And I think we haven't taken this far enough. We're moving, by the way, in case you haven't realized it, into the purely speculative part of the talk where I will give very few results and very few justifications for what I say. The justification here is an analogy to what's worked before. But what other types of data and queries do we have available to us? IGVF is building a gigantic knowledge graph that captures what we know about biology and gene regulation from variants to expression. If we carry that further, we know there are many different scales at which we might want knowledge, and we might want a much bigger knowledge graph for health. There are lots of other data types: wearables data, mobile health, dynamic data. James Jung at Duke and others work with surgical videos, and of course, electronic health records and medical images. I'm going to focus in for today just because I've got some work I can talk about in this space on electronic health records. I should admit that 95% of the FDA-approved predictive models for clinical use are all in the image space, in part because of the success of convolutional neural nets. I'd say even more so because of the objective nature of image data compared to other EHR data. But be that as it may, I'm going to talk about EHR data today, but I'm happy to speculate about bringing any of these others in. So this is kind of a picture again of what IGVF is trying to build. And those existing knowledge graphs can also be brought into this foundation model potentially. But what if we just had EHR data? There are a lot of questions we might want to answer from a foundation model that in the past we've had to handle as one-off machine learning tasks. Prediction, will this person have a heart attack in the next 5 years? But it turns out 20 years ago when I started working with EHR data, I thought we want to know who had a heart attack or myocardial infarction. We just look for the ICD code for that. And it turns out that's very wrong because the code may get entered to bill for a troponin test to see if they had a myocardial infarction or MI, or it may get entered because today's visit's relevant to an MI that happened 5 years ago. And so just predicting, in air quotes, what happened to this patient on this day 5 years ago is a big challenge. Attribution. Why do we believe that this person had a heart attack back then? Or jumping ahead, causal attribution, if you will. Did this drug contribute to that? Did the fact that they were on Vioxx back when that was on the market contribute to the heart attack, or would they have had the heart attack anyway? So a causal phenotype instead of just a phenotype. Personalized treatment decisions, and you can read the rest of these. Adverse events and drug repurposing are isomorphic. It's just whether the event is an unhappy one or a happy one that the drug is causing. And then discovery. Can we actually generate, with generative AI, new molecules? James Zhao at Stanford's doing some exciting work where he takes into account something I had wished we could have done 30 years ago and we were just nowhere near, takes into account not only what legal organic molecules are, but if they're synthesizable. And so he can do active learning, speculate, and generate molecules that can then actually be generated or synthesized in the lab. This is a place where, for example, EHRs and protein language models and protein ligand binding might find common cause. Some of you have heard me give the following example, but I'm just going to give it briefly. I think you all know all this about EHR data, but I'll say when that EHRs are actually not relational databases, although when we get them for analysis, we usually get a relational database pull. But EHRs are based on much older database technology that's optimized for fast transactions and reporting. And so every time we go into the clinic, we get coded, I've got this in words, but most of this would be coded information. And if we'd had an EHR, a few places had one, but if we'd had an EHR when I was in my very first academic job, I just moved back from my postdoc in the UK, it would have been really great for me if right about now you could have said, the next time you come to the doctor, you'll be in atrial fibrillation. Now, atrial fibrillation is the most common cardiac arrhythmia. It's pretty common in people my age, but this was when I was 30 and it wasn't real common. And if I'd not been drinking 6 Mountain Dews a day while programming and taking better care, getting a little exercise, I probably could have avoided it. And so it would have been really nice if you could have predicted it for me. When we get the data, it looks like this: a relational database or data warehouse with tables for demographics, diagnoses, labs, vitals, prescriptions. I ran out of room, but procedures, maybe genetics if your hospital has some genetic data. Another view is just like that time series data I started the talk with. Each patient is a timeline, but typically they're an irregularly spaced timeline. So it's very instructive just when events happen. And so one might also use, instead of dynamic Bayes nets, one might use point processes or neural point processes to model these kinds of data. So one thing that we've already been exploring is to try to use retrieval augmented generation systems, or RAG systems, to analyze EHR data and text about clinical events. And the beauty of a RAG system, if you're not familiar with this, is you can use a smaller language model to embed information that you trust into your query that then goes to a bigger language model that's based on lots more information. Like GPT-4 is based on a lot more text, so that its text it generates is maybe higher quality, but it includes text you don't trust. And a RAG system lets you specialize that to text that you trust. This was some work recently with Jihye Choi, Niels Palumbo, Matt Engelhard, Prasad Chalasani, and Somesh Jha in this year's MLHC, but it was all about using a RAG system to try to extract from trusted text information about adverse drug events. And we'd love to have this access a lot more than the medical text. We evaluated on something I'll mention again later called the OMOP ground truth. Just same OMOP as in the OMOP Common Data Model, Observational Medical Outcomes Partnership. And this is just 10 health events of interest by 10 drug categories. And there's a red here if the drug class can cause the event as an adverse event. There's a green if it helps you avoid it. And this shows the ground truth on the left and the RAG system results on the right. So it's not perfect, but it's getting pretty good. So for example, if you want to use this to evaluate where you don't have a causal ground truth, maybe you've got new proposals for adverse drug events and you want to evaluate how well your system is working, then this RAG LLM approach might work. But if we want to build this kind of foundation model that includes the data that goes into protein language models and includes EHRs and other data types, there are certainly some technical barriers, and maybe I'm being just a little optimistic here, but I think for most of the technical barriers, like the protein DNA binding isn't as good as we want, the solution is wait a year or two. Some of you are working on this, and if you're not, other people are working on this and making great progress. I think the big barriers are social and legal. And many of you have experienced this. It took many years for me to get access to the health data at Wisconsin, even de-identified. Same is true after I moved to Duke. And if I want to share data between Duke and Wisconsin, then it's much tougher. I've got a project with some wonderful collaborators here at UNC and at Wake Forest, and we share code all the time, but we have to run on our own data and not on each other's data. And if we try to do this nationwide or worldwide, you can see the big barriers. Somesh and I and others have worked for years on trying to come up with technical solutions. I think there's going to have to be at least partially a social and legal solution to this. So I just want to be realistic going in on what some of the barriers are to building this kind of foundation model. But I want to give some initial perspectives on the promise for it. This was a vision that my collaborators in Marshfield, Wisconsin, and I had, more than a decade ago, where we said for every patient that comes in, over here on the right, we want to have genetic, clinical, environmental data, and we want to use that to predict future events or response to a treatment we give today for a future condition. And to get that, we're going to take lots of historical data on the left and train lots and lots of machine learning models. Now, the goal would be train one foundation model to do this and also bring in the basic science, the relevant basic science data and mobile health data and so forth. But we did experiments a few years ago with data from the Marshfield Clinic. They had an EHR that's now 60, they've had an EHR at least 60 years, and genetic data on a subset of those patients. They have more than 1 million patients in the record with all the tables that I was showing you earlier. And we did a number of initial tasks like predicting myocardial infarction or predicting atrial fibrillation. We usually tried to predict first occurrence of the event because that's tougher. We usually tried to use age and sex matched controls because, again, that's tougher. And we'd show individual ROC curves, but then we thought, what if we try to predict everything? Eventually, everything meant drugs and procedures, but we focused in on ICD-coded diagnoses. And we plotted the histogram of all the AUCs if we were predicting, distinguishing those who would have the event from those who didn't a month before. And that's this rightmost histogram with an average AUC above 0.8. Next to the left is 6 months, and then in yellow, 2 years. And you see that you're still above 0.7. Because this was Marshfield, we had data going back more than 20 years, so we tried that. At that point, you're almost random guessing, although there is, if you look closely, a small tail to the right, a little bigger than one would expect of events that are predictable. We also said, let's freeze our models and let's get a different set of 10,000 patients and ask what will happen to them in the next year. How well could we predict that? Now it's a much smaller set of patients, much shorter time. So I'm also showing you the curves that would correspond to 95% confidence intervals on these predictions. But things are fairly predictable, and I expect with a proper deep neural network trained on enormous amounts of data and compute power, if we could solve the social and legal barriers, that we could do even better than this. We also found that the genetics didn't help us, but we had it on just a small set of patients, and we already saw a lot of downstream effects of the genetics. So we think we were seeing a lot of clinical effects of the genetics before the heart attack or whatever we were trying to predict that de-separated the genetics from what we wanted to predict. But we did build with de-identified data a method you can download if it's useful to you. Xiaowan Huang led this effort, a method that would build family pedigrees in this de-identified data by saying these two people had the same address for 5 years. I don't know what that address was. And they had the same last name. I don't know what that name was. And you build these pedigrees, and if up above are the results using the clinical data only, the reflection should be the same, except we added some genetic data. We added just, did you have a relative with this event already? And then what degree of relative? And that gives you the shift you see here with significant improvements in AUCs. So I want to leave time for questions. I'm going to spend 5 minutes and leave 5 minutes for questions. I'm going to spend 5 minutes coming back to this problem I mentioned briefly before of causation. Sometimes, like with adverse drug events, all we care about is causation, and hence that OMOP ground truth that I told you about. Other times, we do care about prediction, but even in those cases, the clinicians want to look at the model and say, well, this variable seems really important for predicting that event. And when they see that being on a beta blocker looks like it increases your risk of myocardial infarction or heart attack, that's clearly the result of confounding. And so even if you want to do prediction, if you want the clinicians to trust it, I would argue you want a causal model. And what I mean by a causal model is actually a model that if we go around and just change individual input variables, or if you like, change the distribution the inputs come from, the accuracy will still hold up. That's true of the model that generated the data, but it's almost always wrong with a predictive model we've trained. If you go around and just speculate, well, if I change this person's beta blocker prediction, the model says their heart attack risk goes way down. If you try that, it's not going to work because the beta blocker is standing in for what the doctor believes by examining the patient. This is back to that OMOP ground truth that I told you about, matching drugs and events they cause. This is another picture of that ground truth. The blues are just a sampling of the non-reds, non-greens, for which there seemed to be no literature evidence that the drug is maybe causing the event. The reds are based on actual drug labels, so they're a sound ground truth. OMOP evaluated many, many methods, and propensity scoring methods and other methods work really well often, but in the OMOP evaluation, the best performer was a kind of fixed effect or random effect model where you had patient-specific baseline risks, much like you would have in a Cox regression. And it's just a Poisson regression shown here, but where the B for the outcome O is patient-specific. So XTD is just telling you, is this patient at this time on this drug B? And this is giving you the intensity parameter or the parameter for the Poisson distribution. And that was arguably the best performing model in these OMOP experiments. So to jump ahead to the punchline, I'll say that my student at Wisconsin, Charles Kwong, took this idea to a problem that our collaborator Jamie Thompson had of saying, what drugs can we find that are on the market for other reasons that seem to be lowering cholesterol or lowering blood pressure or lowering fasting blood glucose? And Charles took this idea of a patient-specific baseline further with a time-varying baseline. Now it's a numeric response, so we're doing linear regression. But patient baseline values change over time, as illustrated here with these with the values at these up arrows. And so the first line of the equation is just a standard linear regression. Then on the next line, we start with a lasso penalty. And the last penalty says if we have two measurements at times tau I and tau j and tau j plus 1 for patient I, that are within sigma, let's say 6 months of each other, then we pay a penalty that's how much we change the baseline. So if 100 patients see their fasting blood glucose drop 30 points, after they started the same drug, we are going to put that explanation in the beta for that drug and avoid this penalty. If they all were on 30 different drugs or no drugs at all, we have no choice but to explain the change with the baseline change and pay that penalty. So that's the gist here. To jump ahead, I'll just say it worked remarkably well, and it then worked really well when we took it back to the adverse event problem and the binary outcome. And try to wrap up in a couple of sentences. I'll say what we want to do now, as you can imagine, is bring these patient-specific baselines and even time-varying baselines into deep neural networks, including transformers, for building causal foundation models. There's other work in this space, like causal transformers, but it assumes all the confounders are measured. This kind of approach specifically is aimed at least partially adjusting for the unmeasured confounders. But that approach with the neural network, the approach we used here was to alternate back and forth, like in an ADMM optimizer, alternate between optimizing the model, the beta coefficients, and optimizing the baselines, and you go back and forth. Now, if you're going back and forth between training the neural network and re-estimating these time-varying baselines is completely intractable. There's just no way we can do it. And so what we're trying to do now is use the theory I gave you before to justify putting these baselines directly into the neural network as latent variables. And as you would do in training a Bayes net or Markov net, put not random, small, non-zero weights like you do for a neural net, but really informative weights that say, if this baseline is big, then your chance of an MI, your probability of an MI is big. And if the baseline's big today, it's probably the same next week. And put those kinds of put that kind of prior in to the neural network. And we believe that's justified because we do that all the time in Bayes nets and Markov nets, and we're viewing neural nets now as Bayes nets or Markov nets. So I want to just jump ahead. This gives some of the ideas, but we're trying to also get some theoretical foundations for that piece. But fixed and random effects models lack a lot of the kind of theoretical causal proofs that you would get for a lot of methods like propensity score methods. So we're aiming at that as well. I want to acknowledge my collaborators in the IGVF funding, and I'll put these conclusions up here. And thanks a lot. Any questions?
And
sorry,
could I ask a question?
Slide.
Yeah, ask a questions.
Yeah, go ahead.
Yeah, I think it's a great talk. I enjoyed a lot. I think there's a lot of people working on developing the foundation models for different tasks in the biomedical sciences, right? And you can do foundation model for A, B, C, D. There's many paper on that. And I have a big concern about it because for many data sets and the problem tasks, actually, we don't really have the annotation data sets. And I think that's the big problems in biomedical sciences because compared with a tech company like NLP and the CV problems, for many tasks, you can easily identify the ground truth and annotate them. But as that's not the case for the biomedical sciences in many aspects, because you don't know the ground truth, and you can annotate whatever you like to, but you can claim the names like a biomedical foundation model for ABCD, then so what? And because at the end of the day, and because you still need to go to the treatment and preventions and all the things, how do we really evaluate all these foundation models in one sense? What's your opinion on that?
I got part of that, but I need some of you to help me summarize. Anybody willing to help me summarize that? With my hearing aid, I missed a lot of that.
He's referring to like
Okay, I think unsupervised training in building a foundation model, because you have all the patients across all the time and all the events, I think in effect, as you're doing unsupervised training, you're still really doing supervised training because you're predicting every event that's occurring. So I would argue in training a foundation model like a transformer on all this data that you can argue you're doing a kind of unsupervised training, but it's still getting supervision as it goes. If the concern was, I thought the concern was more about understanding or interpreting where the results are coming from, and that is a challenge. Even if we understand the model as a probabilistic graphical model, it's so large that and often for privacy reasons, we don't get to see the weights. That is a big concern here. So anything that gets proposed by the model, I think would need a human in the loop then to follow up on very closely. So it might be really great for discovery such as drug discovery, but certainly not for unsupervised training or unsupervised treatment of a patient. Supervision meaning something different in this context. There is a lot of interest in model distillation and something called the lottery ticket hypothesis, that if there's a ridiculously large model that's trained to high accuracy, then there's a much smaller model within it that's equally accurate. My hope is that this tree structured view should give us some clues into proving that hypothesis and getting knowledge distillation methods that might help us pull that out. Or the HMC method might give us some other methods besides Shapley values to put in evidence anywhere and ask about any other nodes anywhere in the model. So I'll leave it at that.
I think my point is for CV or NLP like ChatGPT, because I think the data and also computing power is super important. The data is annotated data. But for many problems in the healthcare, we don't really have annotated data. So that for all these tasks, I'm not sure we can really build any foundation models. That's my point.
If the concern is the quality of the annotations, that the EHR data, for example, were entered largely for billing purposes, I completely agree with you. We had a discussion the other day about what can we do with EHR data given how noisy it is. I believe that the whole embedding approach in neural networks, when we go to this smaller latent space, does partially adjust for some of the noise, but I agree with you, it's a big problem. My hope is as we break down some of the legal barriers across sharing the data and start to see some use cases for these models, that will change the whole way we enter the EHR data. But I think that's a social change we need.
Yeah, I think that's a lot of challenges compared with computer vision.
Thanks.
Do you think that this is going to be easier to bring to fruition in a country where you can have like a single payer or a universal healthcare system? So you're not going to have these limitations we have in the US with all the healthcare union disparate systems. So like,
yes,
this.
yes. This is, maybe I'm oversharing here. I don't like to get political, but living in the UK for 4 years started my long, slow march from being the most conservative politically person you could imagine to one of the most liberal. And it was because I realized all the advantages of that single payer system. I was just talking to somebody the other day who was telling me that he started his EHR AI work in Denmark and thought, this is easy. I just get all the data in the country. And then he came back here and saw how different it was. We had our third child born in the UK, and we had house calls from the doctor in the first 2 weeks, and everything was paid for. I won't, I'll get off my soapbox now, but there are a lot of reasons that's a good thing. Yeah.
We're computationally at the point where you have access to the data.
That's right. Yes. And I'm always complaining, even over at Duke, and they'll tell you that, and it's the same everywhere. I've said, to succeed, we need to bring our PhD students and researchers and the data and the high-end compute power together. And I can almost always get 2 of those 3 together, but frustratingly, it's really hard to get all 3 together. Yeah. So all I'm doing is commiserating with you, but I agree. Anything else? All right. Thank you, everyone.
Recording stopped.